Good day, everybody. My name is Nancy Cox. I'm coming to you today from Vanderbilt University here in Nashville, Tennessee. And this is session 1B, genetic maps for M2M2M. And we're going to start out with a really great talk from Hernán Dopazo, who's a population evolutionary biologist and also a really good bioinformatician from the Evolutionary and Population Genomics Lab at the University of Buenos Aires. And he will be talking to us about a big population genome reference that started in Argentina and tell us about the objectives and plans for this. So without further ado, Hernán.
Hello, everybody. My name is Hernán Dopazo. This is a presentation about the National Program of Genomic Reference and Biobank of the Argentinian population called PoblAR. Thank you very much for the International Common Disease Alliance organization to give me the opportunity to explain that. We are interested to show the context, this context that is international and national backgrounds for the presentation of the creation of the PoblAR as the Biobank and the program of genomics in Argentina. We are interested to show you the objectives, mission, and vision, the organization of the Biobank itself, some results and next steps we are producing in this year 22 and the next year 23. That is the objectives and plans. And of course, finally, I will finish putting in context the Latin genomes and ICDA collaboration with PoblAR in the next year. Then, as you know, about a decade ago, the first warning was published about the bias, the great bias of representation in human disease databases, the lack of diversity of the reference panel of genomes of our species, and the risk associated with the possible failure in the application of the precision medicine program for large regions of the world. Of course, the scientific and the public health system of developing countries like Argentina cannot remain invasive. The absence of a national focus in genomic health will produce a strong social inequality, as we know. Here, I show you some relevant publication during most of the decade. Perhaps the very important is that the polygenic risk score, when the predictive accuracy when talking or when using values from Europeans is very, very accurate. But however, when we move far from Europeans, like in this case, Latin Americans, American in general, African, etc., well, the accuracy is lost and lost and lost. As Argentinian, probably many of you know that we are a mixed population of Europeans, of course, but Native American, a great portion of the country is Latin American, with different kinds of ancestry of Latin Americans, and of course Africans too, yes, in minor proportion. Then, knowing this background, this international background, science, 2013, a group of researchers from the national universities, national health system, and the science and technology system too, we meet itself in a consortium. Yes, the common grants of all of us was the ability to collect biological and non-biological information from different regions of the country. We have experience. All the people in this consortium has experience about that in previous years and in different scientific projects, approved and published, etc. Most of us are anthropologists. We know genetics, medicine, informatics, and epidemiologists. Of course, the common interest about all of us is to improve the public health through scientific knowledge of genetics and genomics. Okay. Then, during 14 and 16 years, we produced foundational documents, yes, that were producing different commissions of this consortium that we call PoblAR at that time. These commissions work in the organization and governance of a national biobank. We produce documents about that. How to sample population in Argentina, which are the different ancestries and where are the locations that are important in our country to take into account. What about the information concept that we need to take in a biobank with a degree of a national reference? Of course, we produce a standard operational process and how to work with the incidental findings in the case we obtain genomics data or genetic data. Of course, we talk and produce documents about how to work in the digital part of the biobank, the storage of this biobank data, and the sharing policy. Okay. The working plan and the plan of communication to society is very important for us, and all these documentation were produced in these years. However, we must wait for about 5 years, but in June of the last year, 2021, well, finally, the Ministry of Science and Technology and Innovation of Argentina, well, launched the PoblAR program. That is, today, PoblAR has the status of the national program in the Ministry of Science. This is exactly what we have. This is the web page of the PoblAR program, and this was the event of the launching this program, the presentation to society. As we produce here, you see here the objective of the PoblAR is to design and implement the sampling of biospecimens and associated data from the Argentine population that contribute to the creation of an open data biobank of importance for every or any biomedical and population genomic research in Argentina, and of course open to the collaboration with international groups of research, of course. The mission and the vision is here. You can read after that. It's not sense to talk about that and to read that. But of course, we are not only interested in genomic biospecimen to collect genomic biospecimen, but of course, we are interested in associated digital metadata. That is social data of the population along of the country, and the collection of different kind of data of how the people are living in different parts of the country, and the diets. Of course, we will have and produce information about the scans, etc. The vision, of course, is important, is to consolidate itself as a normalized reference system of multifactorial data for biomedical study in our country using genomics. And of course, we're interested in phenomics too. All this information was written in this national journal, Ciencia, Tecnología y Política. Here is the DOI number, you can download that. And we call this presentation at Genomes, Diseases and Precision Medicine, a national project. Okay, then how is the PoblAR organization? How PoblAR is organized? Well, of course, we have in the biobank, there are two parts. One is the biobank itself, the physical biobank. But of course, we're interested into data analysis and to study the genomic reference and outside the reference population itself. Then we have a part of the governance. We have a part of the biobank structure. The governance is the direction and the advisory committee. The biobank structure is divided in the coordination and administration, the maintenance and quality control of the biobank. We will put people, yes, working in this part of the biobank. Data analysis and storage. This is the easy part because most of the people can do that in Argentina in this part of this kind of analysis. But of course, it's very important that the structure of Biobank contain a communication and the outreach and education program. We will do that. It's very important for us. It's a national program. Then this is exactly what we are testing to do. And the external committees, we have two, of course, external scientific and the ethical, legal, and social committee. But this is the superstructure, but under that is perhaps the more important. The more important is the primary nodes that are distributed along all the country from north to south and the west to east. The primary nodes we have here in the northwest of the country, located in San Salvador de Jujuy, the northeast, localized in the Provincia de Misiones, in the capital of Misiones, Posadas, the center of the country located in the Provincia de Córdoba, in Córdoba City, Patagonia, and this center is basically sampling along all this part of the country. Yeah, the northeast is basically can do the job in this region, and the northeast is all this part. Yes. The Patagonia is located in the city near Rawson. Here, this Puerto Madryn is the same paths, and they can collect all the information, and it's very near in Argentina. Yes, in all the Patagonia too. And the province of Buenos Aires in La Plata node, and the federal district here in the capital city of Buenos Aires. Then we have the possibility to sampling and collect biological and non-biological data for all these countries, not only in a first sample or first pools of sampling, but the idea is to collect data for year to year, and in order to improve the representation of the different ancestries and the different localities of the country along years. About the result we produce, well, I showed you some part of the genomic consortium that different participants of the PoblAR Biobank produce data in genomic consortium. One, perhaps the first one, was the Proyecto Candela consortium. This is a Latin American consortium interested in the admixture and the phenotypic diversity and self-perception of ancestry. They're very, very interested in the data they produce. The data produced by the Latin American Cancer Research Network, Argentina is participating in this consortium too. And then we have disease, we have phenoms, and of course we produce now as PoblAR a consortium itself. Perhaps the best representation of the local ancestry of our genomes in the recent times. Of course, this is the local ancestry picture of a people on a single individual living here in Formosa. As you see, there are a perfect admixture genome of African haplotypes, European haplotypes, and Latin American inbred haplotypes too. One of the interest points was that these Native American ancestries, we know from previous data that we have three different kind of Native American ancestry. That is the subtropical and tropical forest coming from this part of the region of the South American. The Central Andes coming from that, and the Central Chile and Patagonia, yes, coming from to Argentina from this region. However, we find in this population, in this paper, a new. This is an hypothesis today that there are another ancestry not recognized before, yes, in central and western Argentina. This is the work we are producing now. with more and more data, defined scale analysis. Then the next steps, we are interested, this phase 1 creation and funding, we finished that in 2022. We have the biobank consolidation and first sampling. Basically, we're interested in to collect more than 2,500 individuals throughout the different regions of Argentina, collecting biometric social data and to store the biological sample in the biobank, in the PoblAR Biobank. And the genomic studies finally will begin in 2022 with the whole genome sequencing of more than about 100 individuals from the first year. In the phase 3, in the coming in the next year in 23, well, this is the phase 2. That is the development of a strategy of a public-private projects that demonstrate, yes, the cost effectiveness of genomics medicine in Argentina. This is very important for us. There are a lot of people in Argentina that cannot pay an analysis or diagnostic of exome or whatever kind of they are interested. Yes, the medical doctor are interested. It's very, very expensive here. Then we are interested into work in this point, and of course, finally, we are interested too in the international collaboration with Latin Genome. We are part of that, and the ICDA collaboration with different groups with our own genomic data produced from this year to the next one. And of course, this is the strategy of the International Common Disease Alliance, MAPS, Mechanism Medicine. And here I put basically where are the different opportunities that we have here in Argentina from PoblAR or from the National Research Council, CONICET, or the national companies that we have here in Argentina too, that to work in different areas. Yes, of course, PoblAR can do maps and can do produce data infrastructure and collaborate with ICDA in that. But of course, this is a part of the work we are interested to produce in collaboration with another groups, international groups. Of course, with the Latin American Alliance, yes, of Genomic Diversity, that is Latin Genomes, and the ICDA. Well, I expect that I can be clear about what are doing now, and that's all. Thank you very much, all of you, and of course, if there are any questions, I am very, very great to answer that. Thank you.
Hernán, thank you so much. While we wait for some questions to come up, I just wanted to say how impressive it was, that how much planning you guys did in preparation for getting this program started. And I wondered, with all of that, if you had some secrets to share. It looks like a roadmap for sort of best practices in how to get this kind of a study started, and what you've learned along the way. If you'd care to say a few words about that while we wait for a question to come up.
Okay, thank you, Nancy, for this comment. Of course, the work we done was done during many years. Indeed, in 2014, we thought about the initial program of PoblAR and the necessity of the biobank in Argentina that collects genomic data and share this data for all different projects that are indeed doing it in the same country, and of course in collaboration with other institutions around the world too. Then basically we meet together, initially only 4 people, and finally we meet more in the last meeting, well, more than 30 ones. And the idea was to plan exactly how to do this biobank. What are, for example, the organization of the governance? The governance is very, very important. Which are the external committees we need to add in this governance? But of course, the main, I will insist, it's very important to insist that the very basic people are those that are in the country in different nodes, because they have the experience. Yeah, they have the experience for sampling biological samples, and to have they have experience, for example, in the collecting information that is not biological information from health, and making body scans too. That is, we have all the instrument, all the parts that in the laboratory and in the around the country too, in different parts of the country, different cities in the province of Argentina. But we must arrange how to start. Then we decide to write informed consent, POEs, incidental findings, how to inform that, and on the basic policy of sharing data and data storage, and of course the very, very important plan of communication for the people that are interested in this kind of, to participate in the PoblAR. Then finally, after about 7 years, 3 different governments, 6 or 7 different ministries of health, and 3 Ministry of Science, finally we get it. And this is very important. Today we have a program that is important for the Argentina people, important for the people, that the data that we will collect is important for health, and this is a plan that is of course shared between the scientists and physicians, that is from the medical doctors too. Yes.
So I think we have to move on, but I would request that people put their questions in Slack, and Hernán has promised to answer the questions that people put in Slack. And thank you again for the presentation. And we'll move on to our second speaker, who is Saori Sakaue, originally from the University of Tokyo, a physician scientist, who's now a fellow in medicine at Harvard, and will talk to us about cross-population GWAS for M2M2M.
Hi, thank you, organizers, for the invitation for this wonderful meeting, and thank you, Nancy, for the introduction. I'm Saori Sakaue, a postdoctoral fellow at Harvard Medical School and Broad Institute. Today, I'm so excited to talk about our recent research using cross-population genome-wide association study in the context of M2M2M challenge. I think from all the great talks today, which I'm really enjoying, everybody agrees that cross-population genetic study is important, but I will also join the party and explain a bit about this, and then move on to how I try to make genetic useful in medicine, motivated by my question I had in the hospital as a physician geneticist. First, about the cross-population GWAS, it cannot be overemphasized that the genetics maps still need population diversity, where 80% of the GWAS participants are of European ancestry, whereas only 10% of the worldwide populations are Europeans. And so in addition to the European-based biobanks, I was fortunate to work on one of the major contributors to the East Asian data, which is Biobank Japan. So I wanted to analyze and openly share the study results as much as possible to meet the expectation and the responsibility from the community. Just a brief introduction of the Biobank Japan, or BBJ. BBJ is one of the largest non-European biobanks to date, and probably one of the oldest, launched in 2003. BBJ is a nationwide hospital-based cohort of more than 200,000 Japanese individuals. Along with SNP array genotype and whole genome sequence of those individuals, we have deep clinical data, such as more than 200 disease endpoints, family history, age of onset, and lifestyle information. Although the cohort is not directly linked to the National Health Registry, BBJ was rigorously following up participants about their disease, survival outcome, and cause of death. So let's go back to the M2M2M challenge again. So we want to bring this cross-population genetic map to disease mechanisms to contribute to medicine, just as the ICDA has proposed. And I think medicine can be broadly divided into 3 categories, namely prevention, diagnosis, and treatment. So I'm gonna outline my talk based on these 3 categories in medicine. So my first research is about how we can use genetics to identify risk factors of health to prevent undesired health outcomes. And to prevent something, we have to know what the risk is. Maybe polygenic risk, or PRS, is one of the most successful examples of the genetic-based risk prediction. By taking the GWAS effect size, multiplying by individual genome, and summing them up, PRS can now point those with several-fold higher inherited risk of common diseases. But do you think genetic prediction is really useful in prevention of undesired health outcomes? This is the exact question I had in mind when I started this project. The current PRS is mostly used for the individual prediction of the disease onset. But if I tell the patient that she has a higher risk of having Alzheimer's disease in future, for example, she may be confused because the genetic risk cannot be modified and she doesn't know what to do with this prediction. So given this challenge, I used PRS as an instrument to identify modifiable, treatable risk factors affecting our health outcomes, such as lifespan, rather than to predict the genetic risk itself. By associating the PRS of the risk factors with lifespan in population rather than individual, we can identify causal risk factors for health outcome, which is modifiable in anyone. We use PRS rather than the risk factor itself because identifying causal factors have been always challenging in medicine. The conventional observational study, which correlates outcome with the risk factor itself, is always susceptible to reverse causation. For example, low blood pressure is actually strongly associated with shorter lifespan, but we cannot distinguish whether decrease in general health causes lower blood pressure or low blood pressure causes bad health. A randomized control trial, RCT, which medically changed the risk factor value and see the effect on the outcome, is the best way to find causality. However, RCT takes costs and it's not ethically always feasible because that can harm patients. So instead, we used PRS as an instrument to simulate the genetic predisposition to cause the risk factors to identify causal factors associated with lifespan. PRS can be considered to be less susceptible to reverse causation. Given that lifespan is extremely complex trait as an outcome of all possible events throughout our lives, we thought replication and generalizability are essential. So we collaborated with 3 nationwide biobanks from different populations and different health systems, namely BBJ, a hospital-based cohort, UK Biobank, a population-based cohort, and FinGen, a combination of both. This also greatly increased statistical power, which generally depends on the number of individuals who died during the follow-up period. In the biobanks, in total, 670,000 individuals. We associated the PRS of the risk factors with lifespan across populations and discovered multiple drivers of shorter lifespan. I remember I was really excited to see this plot. So those are the risk factors we investigated, and the odds ratios are showing how much they are contributing to the shorter lifespan or longer lifespan. First, high blood pressure causes shorter lifespan by 0.3 years per unit changes. Second, obesity causes shorter lifespan, and interestingly, this effect was much stronger in Europeans than in Japanese. We also find other candidates of shorter lifespan, such as high cholesterol levels and low C-reactive protein. So to summarize this study, we showed the clinical utility of PRS by discovering risk factors of short lifespan, which can be communicated to people and improved through lifestyle modification. So let's move on to my second research, which is showcasing how useful it is to use global genetic map in rethinking about diagnosis. This research has also motivated my experience in the hospital. Prioritizing potential diagnosis of patients, or what we call differential diagnosis, is extremely important to treat patients efficiently. To make diagnosis, physicians have empirically categorized observed symptoms such as fever and cough to describe underlying dysfunction, for example, lung infection, supported by clinical exams such as auscultation. The objective tests, such as plot tests or X-rays, have refined the current disease diagnosis, such as ICD-10 code. Reasonable disease classification is important because that can lead to an optimal treatment, such as to use penicillin to treat streptococcal pneumonia. So I wondered if we add genetic information here, we may be able to reinvestigate or refine disease classifications, which should lead to even more optimized treatment. This is the central motivation of this study. To this end, we conducted a genome-wide association study in BBJ, UK Biobank, and FinGen for comprehensive 220 medical phenotypes. We discovered more than 5,000 novel genetic associations, including Japanese-specific variants. We then constructed a large phenotype by genetic variant matrix about the strength of the association and mathematically decomposed it by SVD, which is similar to PCA, to extract simple genetic components which point to the biological pathways shared among diverse diseases. We also tried to classify similar diseases by using those simple components. This result was very interesting. Here, rows are the derived components and columns are the diseases sorted by the contribution to each of the components. The components from completely unsupervised decomposition were converged on the disease classifications defined by ICD-10, which proves the validity of the conventional disease classifications from genetics. Next, to interpret these genetic components, we took the loadings of each genetic variant per component and projected them onto the GWAS of simpler traits such as biomarkers and metabolomes. This analysis was effective in dissecting complex trait genetics into the combination of simpler traits. For example, this component 2, which was shared among many different cardiovascular traits, was dissected into the more simple genetics of the blood pressure and cholesterol-related traits. We finally classified similar diseases by using the genetic components. There are many allergic diseases such as asthma and atopic dermatitis. We wanted to categorize them into subgroups in a genetically relevant manner because those fine-grained categorization will be useful in optimizing treatment rather than just to use an anti-allergic agents in general. So by taking the composite score from the genetic components, we categorized the similar allergic diseases into two subgroups. The first one is here in red, such as asthma and allergic rhinitis. This component suggests abnormal Ig secretion pathway, which was traditionally regarded as type 1 hypersensitive reaction. The second group is here in blue, such as contact dermatitis and metal allergy. The pathway was IL-13 secretion, which was traditionally regarded as type 4 hypersensitive reaction. So to summarize this, unsupervised decomposition of the disease GWAS surprisingly converged on the knowledge-based conventional disease classifications and had a potential to refine them. So lastly, as our final aspects in medicine, what can genetic maps do for the treatment? For that, I think we finally need the precise mechanisms by which the genetic variant causes the disease. So I would like to change our gear a bit and share my ongoing projects and visions about this challenge. To understand the mechanism of complex GWAS loci, we first typically narrow down causal variants by statistical fine mapping. The functional genomic tracks, such as chromatin accessibility, which is a hallmark of active regulatory region, is helpful in annotating those variants. But one simple yet important challenge is to assign a causal gene to each of the candidate SNPs, especially in gene-dense regions. Although there are multiple methods to answer this, none of them are at single-cell resolution. So my current research is to address this challenge by using single-cell data with simultaneous measurement of chromatin accessibility and gene expression. By directly measuring the effect of epigenetic change on the expression outcome, we can assign a single gene, in this case gene A rather than gene B, to the regulatory region harboring the causal SNP in a specific cell type. This SNP to gene linkage seems to be working very well and may be useful in nominating candidate target molecules for drug development from the GWAS loci with unknown function before doing laborious perturbation experiments. Another important challenge is to fine map causal cell states where the genetic variance causes gene dysregulation, which may not be very obvious when we only look at healthy tissues. In my another ongoing project, by analyzing single-cell data from synovial tissues with rheumatoid arthritis patients, we observed cytokine CCR13's EQTL effect only in the infinite macrophages in rheumatoid arthritis patients. Those causal cell states may be a potential place to conduct target validation experiments. And given that we need to study tissues from different diseases, time points, and organs to fine map causal cell states, we will also need a statistical method to batch correct and combine different functional experimental data sets and analyzing them together to achieve robust and powerful results. Maybe we can think of this in a similar way as we combined hundreds of GWAS cohorts by genotype imputation and meta-analysis. So there are my slight conceptual takeaways from my research. We showed two ways, PRS utility and GWAS decomposition, to make genetics useful in medicine by asking clinically important questions. And for this, I think my experience in the hospital was important. Second, cross-population GWAS is a great resource to answer those questions, to gain statistical power, and to test the generalizability of the complex outcomes among populations. And we will need many aspects of diversity methods to integrate many datasets in functional genomics, maybe somewhat analogous to what we have done in genetic studies. With this, I would like to sincerely thank all the participants of our studies for their invaluable contributions. And I want to thank all the people here, but especially Masa, Mark, Yosuke, Mani for long-term collaboration and analyzing the data together, and all the people at Biobank Japan for hard working for maintaining this Biobank, especially Yuki and Professor Yamamoto, two of my previous mentors, and Shomo and Alkes for advising my current projects, Accelerated Medicine Partnership for the data provision and all the funding sources here. And lastly, we are very open for the collaboration, so please let us know if you are interested in, and all the GWAS results I talked today are already shared at our pb.jp website. And with this, I will close. Thanks for listening.
Thank you so much. And while we're waiting for questions to come in, I think the points you raised on the getting to cell types is a really important one, and one that I think you're right. We have some methods at our disposal that we can use in the context of if we can get enough of the right cell data in the context of tissues to take it even further. And I'd love to hear what your thoughts are on methods for that.
Yeah, that's a great question. And actually, this is one of the main things that I'm currently doing as a postdoc project. So if you only look at the EQTL effects from the healthy tissues, such as GTX data, we might be missing the causal expression changes, which should be existing in the disease patients, but cannot be found in healthy people. So for example, in the rheumatoid arthritis, there should be some pathogenic cells which cannot be in the healthy participants, but should be in the patients. And for those pathogenic cell states, we can only observe the EQTL effect if we study those participants. And we do find many examples of this by first looking at those tissues, and second, to have a method to identify what axis is defining that cell state. So in this case, my example was the inflammatory macrophages and how we define those inflammatory macrophages. And I think those are the exciting place where we can have much more advancement in recent, in those several years.
So thank you. We have to move on, but there are some great questions that got voted up and that have to do with pleiotropy and Mendelian randomization. So please put these in the Slack channel so that Saori can answer at her leisure. And we'll move on then to the last talk. So for our last speaker today, we have Daniel MacArthur with us now from Australia. We were used to having him here in the US, but all Australians want to go home at some point. Daniel is now the director of the Center for Population Genomics. He's a population geneticist and a really thoughtful scientist on variation in general populations. And he's now working with this Center for Population Genomics that's a partnership between the Garvin Institute and the Murdoch Children's Research Institute. So Daniel's going to talk about the Australian experience with getting more equitable genomics into population studies.
Thanks very much, Nancy. Great to see you and to be part of this this very exciting meeting. And looking forward to more discussion tomorrow as well. So today I wanted to start my presentation by acknowledging that I'm presenting from the traditional lands of the Wangal people of the Eora Nation, and I pay my respects to their elders past, present and emerging in the spirit of reconciliation. What I wanted to do today is to talk through firstly some prior work that I had done while I was previously based at the Broad Institute, particularly discussing very briefly some of the work done around the EXAC and NOMAD projects, and then segue into the work that I'm now doing as part of this new centre, which Nancy mentioned, the Centre for Population Genomics. I will warn in advance that the projects that we're doing as part of the CPG are still at a very early stage. So the last part of my talk will be very much promissory, talking about projects that are in early stage and still in progress and won't be showing results. But hopefully at least some of those projects will be of interest to the ICDA community. So first, just a quick recap of work that's been done over the last 8 years at the Broad Institute in the general area of building large databases of genetic variation. These occurred in 3 large waves at the Broad, with more waves to come. The first of these was the release of the Exome Aggregation Consortium, or EXAC dataset, back in October 2014, which comprised just over 60,000 exomes. A couple of years later, our team was able to release another dataset called NOMAD v2, which contained both exome and, for the first time, whole genome sequence data. And then about 3 years after that, we released the NOMAD v3 dataset, which was the first large-scale whole genome sequence release on the new GRCH38 reference genome. So these datasets in total represented large collections of aggregate data that were primarily put together to provide a reference dataset for the clinical interpretation of genetic variants found in rare disease patients. A few key points about these datasets. The first is that they required shifting and processing massive amounts of raw data, more than 10 petabytes in total across the 3 different waves, spanning over 200,000 individuals. And for the latter 2 projects, that work was all done. All of the processing analysis was done in a cloud-based framework on Google Cloud, and largely thanks to the work of the data sciences platform at Broad and the Hale team, also at Broad. The EXAC and NOMAD projects didn't generate their own sequencing data. We worked with consortia that were generating those data, including many people here as part of the ICDA, who generated those data as part of case control studies of adult onset complex disorders, with many of the big hitters being in the cardiometabolic space and neuropsychiatric conditions as well. Because these were case control studies, we were able to often identify the individuals who had severe pediatric disorders and remove those individuals from the data sets. The reason these were disorders like, for instance, autism, early onset pediatric cancer, or other known Mendelian conditions. And the reason we did that depletion was to try to come up with a reference data set that was as representative as possible of the general population. All of those data were processed and jointly variant called, and as best we could, although this was limited, as I'll come back to later, as best we could, we focused on accumulating diverse range of ancestries. And although the dataset was still dominated by European ancestry, almost 60% of the individuals were of European ancestry, we were able to obtain in Nomad v2 more than 10,000 samples apiece from South Asian, Latino, African or African American, and East Asian populations. For anyone who hasn't already seen the Nomad browser, I'll just briefly mention that all of the Nomad variants and raw downloads of the variant lists that we've identified are available at nomad.broadinstitute.org. Because that's been made available openly and is such a large resource, it's really heavily used, including in effectively all clinical genetics pipelines worldwide. And all of the variant data can be accessed on all three of the major cloud vendors if you're interested in accessing that directly on the cloud. So it's interesting to think about where these large-scale genetic aggregation databases will be going. And here I think there's a few points worth making before I turn to the Australian situation. The first key point is that it's clear that there will be the opportunity for massive increases in sample sizes over the coming years, and including the Nomad v4 release, which we hope will be released in the second quarter of this year, which will span somewhere, we believe, over 700,000 exomes. And that is a remarkable increase, about a fourfold increase over the sample size we've had previously for exome data. And that provides a whole host of scientific opportunities, particularly in terms of increasing power for the detection of constraint, of variation level constraint. But that's not the limit. There's an opportunity for millions more individuals to be aggregated over the next few years. And this includes really amazing projects like the All of Us project in the US, for instance, as well as other large-scale academic efforts where data sharing is built into the philosophy from the outset. But it is worth noting that at some point, the aggregation of genomic data will begin to slow as we start to hit the point where increasingly larger fractions of data are being generated outside academia in settings where data sharing is not the key priority. So in, for instance, industry and healthcare settings, or settings where there is and I'll come back again to this later, where there are geographical restrictions on data sharing that will require a federated approach to making those data available. And then another thing we'll need to think about as we work on projects like Nomad over the coming years is increasing our focus on ancestral diversity. That's something that's already been a recurrent theme, of course, today. That's something many of us are thinking about in the genomic space, but it is going to require significant deliberate effort because Nomad is fundamentally dependent on the generation of data from other groups. So this really requires the genomics community as a whole to be pulling in the direction of greater diversity. Now, that diversity has been very much on my mind in the years that I spent leading the Nomad project up until 2019. We thought a lot about the inclusion of ancestral diversity. That's still a big focus of the project. But one of the big frustrations for me was that for many of the communities that live in my native Australia, there is not yet good representation in large reference databases like Nomad. And that's a consequence of most of the sequencing being done in the UK and the US, which of course are also very diverse communities, but with a very different slice of international diversity than what Australia has. Australia is an incredibly diverse nation. Over a quarter of us were born overseas. More than 50% have at least one parent born overseas. We have a large Indigenous, Aboriginal and Torres Strait Islander population, and also many other communities who've arrived from all over the world in over the last 200 years. Because that diversity is not reflected in large databases like Nomad, at least for many of these communities, there's real impact on rare disease patients who are drawn from these Australian communities. They do not have good representation, and as a result, they're more likely to have variants of uncertain significance and less likely to get an accurate diagnosis than those of European ancestry. So that was the incentive behind the creation of the Centre for Population Genomics. As Nancy mentioned, a joint venture between the Garvan Institute of Medical Research in Sydney and Murdoch Children's Research Institute in Melbourne. The core principles of the centre is that we build datasets in respectful partnerships with communities. We focus on diversity in our research subjects as well as in our team. We do everything as much as possible in the open, including all of our code being open source, our data open as much as possible within the constraints of participant and community wishes. And we build our systems upfront for scale with an eye towards the hundreds of thousands of genomes coming over the course of the next few years. I'll skip very quickly through the the arrangement of the centre, but very briefly, we have a set of foundational platforms that we've been developing around engagement, recruitment, dynamic consent, and return of results, as well as cloud-based computational platforms, largely developed in collaboration with the Broad Institute, particularly the data sciences platform and the HAIL team. And then on those foundations, we have a set of scientific projects. I just wanted to mention two broad areas today. The first is the creation of a more representative database of genetic variation in the Australian setting. And then the second is the application of genomic data to large existing cohorts with clinical and preferably with functional genomic data. So I'll just give a few quick snippets of projects that are currently underway in the Australian setting that we're involved in. The first in the Indigenous genomics space has been a massive learning experience, I think, for all of us over the last 18 months of the centre's development. Working with Indigenous data, of course, requires a fundamentally different approach to engagement, to the governance and the sharing of data. And as a result, we have been very fortunate to be able to join now a large Indigenous-led national consortium that is working to establish a new framework for Indigenous genomic data management in the Australian setting. And this is led by two fantastic Indigenous research leaders, Alex Brown and Azure Hermes. The centre is currently in the process of coordinating the sequencing of the first large-scale genomic dataset from this collaboration. This will be 1,200 genomes of Indigenous individuals currently living in South Australia. And there will be a community engagement and pre-consent process to allow that to be built into a public reference database. We also have long-term plans ahead. We and others will be doing long-read sequencing on a significant number of individuals. And the long-term, we plan to build a representative national cohort of Indigenous communities with genomic and clinical data that can be used for both clinical and research use. But within the constraints of permitted usage, including oversight by an Indigenous governance committee. Outside the Indigenous space, we're working on building a more representative database of more recently arrived communities within Australia. We've gone through a fairly deliberate process to get this up and running. The first of these steps has been identifying the communities in greatest need of increased representation in genomics. To do that, we've gone through existing census data, a lot of existing published work on the distribution of ancestry within Australia, and also performed a survey of clinical geneticists and genetic counsellors across Australia. That's allowed us to identify a set of communities within Australia who desperately need increased genomic representation. In fact, what we found is that nearly all of the large non-European communities that live in the Australian setting are either underrepresented or in many cases entirely missing from large reference databases like Nomad. That means that there's at least 3.5 million Australians who currently do not have good existing reference data that could be used in the context of genomic medicine. These include regions from Oceania right the way through to the Middle East and Sub-Saharan Africa. To work on engagement with these communities, we've developed a team that is performing co-design with each of these groups to develop a strategy for engagement and recruitment for each of these communities. That's being led by Maya Ambagalka in my team, along with Sam Croy, an anthropologist, also working with Maya. And we've benefited here from an amazing partnership with the Australian Red Cross Lifeblood, a blood donation service, which shares our goal of increasing the accessibility of blood from a diverse range of communities, in their case, to increase the representation of rare blood types in the donor community. Our goal is to recruit 10,000 participants initially from 10 communities, with a focus then on improving rare disease diagnosis, but then establish the foundations that will allow larger scale recruitment from each of these communities to allow additional study designs, such as the calculation of PRS, for instance. We'll also be collecting DNA for whole genome sequencing, as well as cells for single-cell RNA-seq. And then we have established pathways now for the return of clinically actionable findings to participants, which is more challenging in many of these communities. The sharing of individual level and variant frequency data, and also genotype-based recontact. In terms of what we'll be able to do with that combined data set of genome and single-cell RNA-seq data, I suspect for many of the people in this audience, they won't need convincing that this is an exciting data set. But we have been working on building some pilot data sets to really explore the analytical methods in this space. Right now, these will consist by the end of this quarter of 1,200 individuals with whole genome sequence data, as well as single-cell RNA-seq from PBMCs. And this is work that's largely been in collaboration with Joseph Powell at the Garvan Institute, who's been driving all of the single-cell RNA-seq work, and we've been working on getting the genomes up and running. The major focus areas here will be looking at novel variant types, rare coding and non-coding variants, structural variants, short tandem repeats, and somatic variants, and their contribution to cell type-specific gene expression. So we'll have that data generated by the middle of this year, and looking forward to getting stuck into the analysis of that. Then the final project I just wanted to mention, because of its scale, but also its inclusiveness, is the Generation Victoria, or Gen V project. And I won't take credit for this. This is a project that's taken many years to get off the ground, largely thanks to enormous amounts of work from Melissa Wake. The goal of Gen V is ambitious. They are approaching every baby born in the state of Victoria over a 2-year period. That's estimated to be just over 150,000 babies, to ask them to participate in a birth cohort study. Their target is to reach over 100,000 families by the end of 2023. And so far they're getting about over a 60% consent rate and climbing, which suggests they will hit that target. They've now hit 11,700 babies and over 30,000 participants in total in just a couple of months of collection. They'll have biosamples from the babies as well as parents, linkage to EHR data, as well as additional phenotype collection. And one of the reasons I mentioned Gen V here, in addition to its scale, is the fact that they have invested heavily in improving inclusion of ancestry of ancestral minorities from the outset. And that's well reflected in the early data. So in those 30,000 participants recruited so far, the ancestral distribution is an eerily close match to the population prevalence of different ancestry groups, suggesting that they are actually managing to recruit evenly from across these different groups. We have 2,800 participants whose primary spoken language at home is not English, spanning 61 different languages, testament, I think, to the work that's been done in translation and interpretation in the consent setting. So that, I think, will be a really exciting project because of its study design, availability of trios, scale, and the availability of phenotypes in that early pediatric period. We're now seeking funding for cohort-wide genotyping array and genomes. So just a final slide about some of the challenges ahead. Of course, there is, although I think we have an unusually productive group that's now focused on solving the challenges around engagement and data governance and sovereignty for Indigenous genomics, there's still uncertainty ahead around how communities will engage with this effort, the degree to which participants will be willing to join these large-scale efforts, and exactly how we manage that data long-term. Australia still has limited experience with large-scale genomic data management. I think we've been able to benefit, I think, in many ways from watching the lessons learned by other large-scale projects moving ahead of us. There's limited funding for genotyping and sequencing, which I suspect is an internationally uniform problem. But then there is one problem, one challenge that I think has been quite interesting to me, is the unusual conservativeness of the Australian research community around return of clinically actionable findings. And I think there will need to be a major cultural shift in that space in Australia to encourage participation of individuals in these types of large studies, given the increasing expectation that results will be returned in that setting. So that's something we're building into all of our studies, and I hope will become more pervasive in Australia over time. So with that, I'll finish by thanking everyone who was involved in developing the NOMAD dataset, and particularly massive thanks to Mark Daly and Heidi Ream, who now co-direct the NOMAD project following my departure from Broad at the end of 2019. Thanks to all the PIs who've contributed their data to that project. And finally, thanks to the Centre for Population Genomics, who've been building the infrastructure required to get all of those genomic projects up and off the ground. Thanks very much.
Thanks so much, Daniel. I think we have to go on to the wrap up from Mark and Cecilia, but please put your questions in the chat, in the Slack, and Daniel will answer.